Ever wondered about the secrecy of cloning? Today, genetic replication is being broken down into digestible chunks. Along the way, we'll also take you on a tour of the top 5 places that have been both secretly and publicly involved in cloning research. Firstly, what is cloning? In essence, it's the process of producing identical individuals, be it plants, animals, or even microorganisms, through non-sexual means. The journey of cloning starts with the extraction of a somatic cell, which is any cell of a living organism other than the reproductive cells. This cell contains the complete genetic blueprint of the organism it's taken from. As we navigate through the science, let's take a detour to our first stop, the Roslin Institute in Scotland, known for creating Dolly the sheep, the first cloned mammal. Next, we head east to the Riken Center in Japan, a hub for pioneering research in primate cloning. Third on our list is the controversial Clonaid in the USA, which claimed to have cloned a human child in 2002. Following this, we have the BGI in China, a leader in genome sequencing technology and active in cloning research. Lastly, we visit the Seoul National University in South Korea, where the first cloning of a dog took place. Returning to our journey through cloning, the second step involves obtaining an egg cell, also known as an oocyte. This oocyte has its nucleus, which contains its own DNA, removed to create what we call an enucleated egg. Now here comes the magic. The somatic cell and the enucleated egg are fused using an electric shock. This fusion results in an entity that has the potential to grow into a full organism. It's essentially a fertilized egg, created without the need for sperm. This entity, or embryo, is then implanted into a surrogate mother. If all goes well, this surrogate mother will give birth to an organism that is genetically identical to the donor of the somatic cell. Now, on to the intriguing question. How can you tell a clone from its original? It's not as straightforward as you might think. Here are 10 key factors to consider. 1. Age. Clones are born at the age of 0 but carry the genetic age of the donor organism. 2. Telomeres. These are the protective caps on the ends of chromosomes. In clones, they are typically shorter than in naturally conceived organisms. 3. Mitochondrial DNA. This type of DNA is only inherited from the mother. Clones will have the mitochondrial DNA of the egg donor, not the genetic donor. 4. Epigenetics. This refers to changes in gene expression. Clones and their genetic donors will have different life experiences, leading to different gene expression. 5. Mutation rate. Clones tend to have a higher rate of mutation. 6. Phenotype. Despite identical genetics, clones may not look exactly like their donors due to environmental factors. 7. Health issues. Clones often have more health issues than naturally conceived organisms. 8. Birthmarks or scars. These are not genetically determined and won't be passed on to the clone. 9. Fingerprints. Even identical twins have different fingerprints, and so do clones, and 10. Personality. Just like with identical twins, clones can have different personalities. In conclusion, cloning is a complex process that involves the fusion of a somatic cell and an enucleated egg, followed by implantation into a surrogate mother. While clones are genetically identical to their donors, there are numerous factors, from telomeres to personalities, that distinguish them from their original versions. So the next time you hear about cloning, remember the intricate dance of science that makes it possible, and the myriad factors that ensure no two organisms, not even clones, are truly identical. Often, a question that arises is, do clones walk among us? It's a question that sparks intrigue and apprehension alike. The current state of cloning technology, as we discussed earlier, allows us to clone animals with a fair degree of accuracy. However, the technology is far from perfect, and the process is fraught with complications, making the possibility of successfully cloning humans a distant reality. Moreover, the ethical and legal considerations surrounding human cloning present additional hurdles. Cloning involves creating a genetically identical copy of an individual, raising questions about identity, consent, and a host of other ethical dilemmas. Legally, many countries have laws in place that prohibit human cloning, further limiting the possibility of human clones existing among us. Even if we were to overcome these scientific and ethical barriers, the probability of clones walking among us would still be incredibly slim. The process of cloning is not only complex and expensive, but also highly regulated and scrutinized. So, while the science of cloning has advanced significantly, the existence of human clones among us remains a topic of debate and speculation. It's a fascinating concept, but one that, for now, remains firmly in the realm of science fiction.